This interview is for information only and should not be considered as investment advice or a recommendation to buy shares in the company featured. Welcome to this Stock Box interview.
Joining us today is Allan Syms, the CEO of Cizzle Biotech, following an announcement of the expansion of its current research program with the University of York to develop its Ciz1b biomarker technology for early stage cancer diagnosis. Well, thank you very much for joining me again today, Allan. It's been a little while since we've spoken, but how are things going generally? You said earlier you've been pretty busy these last few weeks.
Very busy, not just the last few weeks, but throughout the year. And, particularly poignant this month, August the 1st was World Lung Cancer Day. And that day is really designed to bring awareness and bring some education to the process because, it's a fairly scary disease we're talking about. And, one of the key things for me is that, today, 24-hour period, 5,000 people will lose their lives because of lung cancer. So it's such an important topic that, we take pleasure in beavering away at bringing a solution for early diagnosis. So yes, so it's been a very busy period.
Okay. I wonder if you can explain a little bit more about the decision to expand the program here with the University of York. What factors contributed to the decision to continue and how will it work?
Well, first off, we should say the home of the company's research and development is the University of York. Professor Dawn Coverley, that's where she discovered this aberrant protein that's highly associated with lung cancer. So it's really, her leadership and her team that have done all of the work looking at this Ciz1b biomarker. All of that work has been done at the University of York. And we, as Cizzle Biotechnology, we're a very sort of light company in terms of overhead. We don't have our own research labs, but we don't have our own overhead that goes with that. That means that, we are very light in terms of cash burn, and we contract out everything. We're almost a virtual company. And that was a very deliberate decision because our view was we could not hire all the experts that are in the University of York. We couldn't hire the best experts to do production and manufacturing. We couldn't hire the best people to do the regulatory part. So we should contract that work out to the best people that do that. And in terms of the stage of where the business is right now, the very best people to do that are actually in Professor Coverley's laboratory. So number one is that's the best place to do this work. And secondly is the timing of this expansion. We've had this relationship with the university since the company's inception, since the discovery of the biomarker. So for us, we've always supported and invested that research and development program. But in the last 6 months in particular, there's been some really exciting developments. And so we've decided to really increase that investment and really push forward now with the work that we're doing within York.
And how does it work with a partnership with a university like the University of York? You mentioned there about having very light overheads, and as I understand, Cizzle is a spin-out from the University of York. Of course, a lot of the research these days is done in universities and then they spin out into public companies for funding, I guess. But how does a partnership with a university work? Are you effectively contracting Professor Coverley's time and resources?
Well, crudely, yes, we are contracting her time because at the end of the day, all of the intellectual property that arises from this research belongs to the company. So we are doing that, but it is not a simple contract situation. The situation is very much a partnership. It's very much a collaboration. And Professor Coverley has a foot in both camps. She's on our board of directors. She runs and is head of our science program. And she has a day job, which is to do cutting edge research at the university. And from our perspective, we would have to invest millions of pounds to achieve the same state-of-the-art equipment that is in the university. And so through Professor Coverley, we get this fantastic access, state-of-the-art facilities, fantastic, highly trained technical and scientific resource. So it puts us in a very, very good position to make the groundbreaking sort of discoveries and advancements that we're actually able to do. And that's very cost effective for the company. gives us a real shop window to everything that's going on in the latest area of lung cancer research.
Okay. Well, you mentioned earlier about a breakthrough. I wonder if I can ask you how things are going. Clearly, they're going well. If there have been breakthroughs and you're looking to expand the research program here, can you give any of the key things that perhaps have given you and the team lots of confidence to continue pressing on?
Yes, well, look, the first thing is this is a very hard thing to do, and the reason being, and I'm not going to make this technical for the people listening in on this podcast, but if you can imagine that this protein is a bit like a ball of string, and the aberrant protein has a bit missing in it. So, in other words, if you wrap it all up into the ball, the way I like to refer to our aberrant protein is that it looks a bit like the Pac-Man. In other words, there's a cheese bit missing.
Okay.
And our difficulty is that the bit that we want to detect is at the back of the Pac-Man's throat. So to be able to detect it, we have to unravel that ball of string. That's quite a difficult task. We have to unravel it all. And then we have our particular biomarker detectors, these so-called monoclonal antibodies. And these antibodies will detect the bit that's missing in the back of the Pac-Man's throat. And that is technically very challenging. And to do that, you have to have a range of these very difficult to produce monoclonals. So the first thing that's really happened is that we've always had antibodies. We've always had a monoclonal. Now we have a whole range of these antibodies. And the reason why we can now detect these is that we've changed our basic platform from being the research platform into now being what will become the commercial platform for doing regular CIS1B biomarker testing.
Well, I think that is important, isn't it? You mentioned this in the RNS about commercializing the product. The next steps are to finalize standard production and operating protocols and to commence clinical trials that should lead to an accredited laboratory development test and launch initially in the United States. So that's quite a nice, significant statement there to be making. So what are the next specific steps that you're taking on the timeline to get through all these processes and trials to get to the regulatory approvals?
Right. Well, so let me just first clarify. These aren't the first clinical trials that we're doing. We've done quite a lot of clinical trials with very successful results. But that was all done on the research product, if you like, the proof of concept product. So all of those clinical trials now need to be repeated in the commercial platform. And the commercial platform for us was in the lab. It's all very well if you've got top scientists that can do great research and make these results work. But to make that scalable, to make that a commercial product in a pathology laboratory, what you really want is a machine where you can take your blood samples, pipette them into small tubes, put it into the machine, walk away from the machine and come back a few hours later with all the results. And that's the big breakthrough. That's what we've been able to do, is that we've gone away from the basic hard work of a research project into a platform, as we call it, where you can effectively take your blood samples, put them in the device, come back later and read your results. So you now have to repeat those clinical trials on that platform. And that will be the platform that will get approved. Now, the regulatory area is a very complex area as well. Obviously, we need a test that has been very well proven, that it won't fail, that it will get the results correct most of the time, if not all of the time. And to do that, you therefore need to conduct and design these trials very carefully. So we've also referenced that we've got a new partner in the clinical regulatory area that is helping us design those trials. And the reason why the United States is that there are various types of regulatory approval that you can get for a product. The shortest one, the one that will take us to market the closest, is known as a laboratory developed test. It is a prelude to the standard FDA required 510K test that everybody refers to when they take their kit to market. But this is basically taking it to an accredited laboratory, and in our case, CorePath Laboratories are our partners in the USA. CorePath is a specialist oncology pathology laboratory that takes blood samples from 22 states across the United States and brings that in for analysis. And so this product would fit within their range. They would then do those clinical trials, they will do those tests. And at the end of that, they will satisfy themselves that this is giving a reliable, reproducible, robust outcome. So that is the next step. And that's why we're doing this in the USA, because you're allowed in the USA to have these so-called laboratory developed tests, which means provided they're being run in an accredited laboratory, such as CallPath, then this will get you the earliest point to market.
Okay. So I understand what you're saying, I think. So you've effectively managed to commercialize the getting the test result, which is important rather than taking a long time. You've managed to get that to a commercial stage, but it's automatic almost with a machine. The next stage is to basically confirm the reliability of the test and the outcome that is obviously needed. And this will be done with these people in the United States. So what are the next steps then? What will Dawn be doing? We should be working with partners in the United States and running these tests for the next few months.
Well, not only do we have more antibodies, which will actually sort of be interesting to see how they all perform within this environment. So we'll be looking at all of that. Then you get into this whole process of stability. How stable are the compounds? Can you sort of store them at room temperature? Do you have to freeze them? There's a lot in terms of batch and quality control of reagents. So a lot of work will be done in terms of optimizing the way in which these will actually be served, they will be produced, and how they will be sort of sent around the world. And of course, it's not just the USA, because once we get to that point, we will then broaden out our markets. We've also talked to you previously about our interest in China, and that will also be a huge market for us. And the British government, through their embassy in China, is hosting a roadshow, which we've been invited. So I'll be spending a few days in China next September, well, next month, in fact. So to go and meet other corporates, investors, people that are interested in solving what is going to become a very, very large problem for China as well. So Dawn's role is not only just to provide this in a form that's a reliable and robust means, but she's also going to be knocking on the door of the next product. And that will be for another interview. Yes. But we have indicated before that our ambition is to turn this into a point of care test. Our ambition effectively is to make a test just the same way that people test for COVID. We want a test like that, where you can do the same thing for CIS1B, so that a doctor's office, a pharmacy, or even at home testing could be possible.
Okay.
The idea of using a COVID-style lateral flow device. So she'll be also looking at that next part, and we've got a lot more to tell you about at the right time.
Indeed. Well, that does sound like it could be quite exciting when that story can be told in a bit more detail. Just a final thought, Alan. If you were to set out the next immediate steps, let's go perhaps towards the end of this year, so the remaining sort of 5 months of this year, where will the activities be focused? But also, I noticed that you funded until 2024. So how long does your sort of funding last for to take you through these next few steps?
So number one is scale up. Obviously, we've got research level quantities we now need to scale up. So a lot of the work is currently being used to scale up the amounts that we will need for commercial launch. Okay. So that scale up process is first. Once we've batch controlled those, that's the material we'll use in the clinical trial. And at the end of that process, then particularly in the United States, we'll be looking at the reimbursement codes that are needed to actually pay for that test. But we've already started work. These things aren't going to happen serially. They happen in parallel. So we've already got an indication of what those reimbursement codes, but that all leads up to being able to then launch, commercialize, charge, and gain revenue for the business. So those are the priorities there. In terms of cash, you're quite right. We're funded through to the end of 2024. The market conditions right now are very, very poor indeed. So we're actually in a very good position of being well funded right the way through to the end of 24. So, the company's in a very good financial position right now.
Okay. Well, thank you very much for giving us that update, Allan Syms, CEO of Cizzle Biotech, and I look forward to catching up as the story further develops.
Chapter 2 coming up.
If you enjoyed this interview, then give us a thumbs up, a like or a retweet. subscribe to us on YouTube or follow us on Twitter and hit that notification bell to be the first to know when we release new content. There's loads of great content on our website too, across all our programs at stockboxmedia.com. Thank you for watching.